TIMM8A, MATE-A, mitochondrial import inner membrane translocase, subunit TIMM8A, also known as Deafness-dystonia peptide or protein, is an enzyme that in humans is encoded by the TIMM8A gene. This translocase has similarity to yeast mitochondrial proteins that are involved in the import of metabolite transporters from the cytoplasm into the mitochondrial inner membrane. The gene is mutated in Deafness-dystonia syndrome or Mohr-Tranebjaerg syndrome, MTS/DFN-1, and it is postulated that MTS/DFN-1 is a mitochondrial disease caused by a defective mitochondrial protein import system. Structure: the TIMM8A gene is located on Q arm of chromosome X in position 22.1 and spans 3,313 base pairs. The gene produces an 11K protein composed of 97 amino acids. The structure shows resemblance to yeast translocase of the inner membrane TIM proteins with two conserved paired cysteine residue motifs. The cysteine residues organize zinc ions for stability and control other interactions with proteins. Function: the human TIM8A gene codes for a translocase involved in the import and insertion of hydrophobic membrane proteins from the cytoplasm into the mitochondrial inner membrane. It is also required for the transfer of beta-barrel precursors from the TOM complex to the sorting and assembly machinery SAM complex of the outer membrane. It acts as a chaperone-like protein that protects the hydrophobic precursors from aggregation and guide them through the mitochondrial intermembrane space. The TIM8-TIM-13 complex mediates the import of proteins such as TIM-23, SLC25-Atwell/Arula-Rhone and SLC25-A13/Arula-R2, while the predominant TIM-9-TIM-1070K complex mediates the import of much more proteins. TIM8A has been implicated as a required element in normal neurologic development. Clinical significance: mutation of TIM8 is associated with Mohr-Tranebjaerg syndrome/Deafness-dystonia syndrome MTS/DDS, a mitochondrial disease postulated to be associated with a defective mitochondrial protein import system. Mohr-Tranebjaerg syndrome is a recessive, X-linked neurodegenerative syndrome characterized by early-onset deafness, followed by progressive dystonia in adulthood, progressive sensorineural hearing loss, mental retardation, dysphagia, paranoia, and cortical blindness. It is known to be caused by a truncation or deletion of the 11K protein product of TIM8A. Defects in this gene also cause Jensen syndrome, an X-linked disease with opticoacoustic nerve atrophy and muscle weakness. A 39-year-old Japanese male patient with a nonsense mutation of the CGA codon 80 of exin 2 by TGA in the TIM8A gene was diagnosed with Deafness-dystonia syndrome. Signs and symptoms included sensorineural deafness, dystonia, blepharospasm, brisk deep tendon reflexes, and personality changes. However, there were no visual or sensory disturbances. The mother was found to be a heterozygous carrier for the mutation. Another patient, an 11-year-old Dutch child with a de novo missense mutation C66WC233CG in the TIM8A gene, impairment associated with deafness-dystonia syndrome. Signs and symptoms included hyperreflexia, dyspraxia synkinesis, atrophy, and progressive dystonia. A third patient, a 30-year-old male with deafness-dystonia syndrome, was found to have a novel 108G mutation in the TIM8A gene. Signs and symptoms were generalized dystonia. There are many more cases of mutations in the TIM8A gene with varying symptoms commonly, including dystonia, mental deficiency, sensorineural hearing loss, optic atrophy, and others. Interactions. TIM8A has been shown to interact with signal transducing adapter molecule and TIM13. 3 copies of TIM8 and 3 copies of TIM13 assemble to form a 70K TIM8 TIM13 complex with heterohexamer structure in the intermembrane space. The TIMM8 TIMM13 complex associates with the TIMM22 complex, whose core is composed of TIMM22